Good afternoon, everyone. Thank you for joining us today for our weekly discussion on weed science and research. This week we'll be addressing herbicide resistance with alternative chemistries. We'll get started here in probably about 45 seconds. Again, thanks everyone for joining us. I'd like to introduce Dr. Stanley Culpepper, WSSA President, to kick things off for us today. Dr. Culpepper.
Thank you so much, Eric, and thank you everybody for joining the Weed Science Society of America and USDA-ARS as we offer the 7th webinar in this unique series. My name is Stanley Culpepper, and I'm the current president of the Weed Science Society of America. For those of you not familiar with how this webinar series came to be, Dr. Steve Duke and Steve Young submitted a symposium request to the WSSA board to be shared at our annual meeting this past February. It, of course, received full support for our board. When the annual meeting unfortunately had to move to a virtual format, we decided to offer those approved and supported symposiums throughout the year through our webinar series. Today, again, is our 7th with USDA-ARS. So now it is my privilege to welcome you and to introduce Dr. Steve Young as your moderator. Steve is a weed scientist and the National Program Leader for Weeds and Invasive Pests at ARS. Steve?
Thanks, Stanley. Appreciate that introduction. And again, I appreciate the support from WSSA to host this webinar series. And as you mentioned, this is number 7. We've been making our way through several different topics, several different themes. And today we're going to wrap up with the theme of mechanisms. And we're going to hear about herbicide resistance. So I want to introduce our speaker, Dr. Scott Baerson. He's got 32 years of research experience and is widely recognized in the allelopathy and pest resistance fields. He has pioneered the use of genomics-based approaches for studying plant root hair cells, and along with his ARS colleagues, elucidated the full genetic and biochemical components of a novel allelochemical biosynthetic pathway, representing a major contribution to our understanding of plant secondary metabolism. He is currently leading the efforts to deploy this pathway for the development of transgenic crops more resistant to weed infestations. Dr. Baerson also made significant contributions to the Roundup Ready brand as an industrial scientist at Monsanto through his work in the gene expression, weed resistance, and specialty crops biotechnology fields. During the course of his career, Dr. Baerson has also been awarded 11 patents, has had his work appear in numerous prestigious journals, including Plant Cell, Plant Physiology, New Phytologist, and PNS, and was a recipient of the prestigious Grodzinski Award from the International Allelopathy Society. So with that, I'm going to turn it over to Scott, but first I'm going to remind folks, this is supposed to be a discussion, supposed to be an engaging event, so please use the chat box or the Q&A box to put your questions, and we'll get to as many as we can after Scott's finished with his presentation. So Scott, go ahead and take it away.
Okay. All right. Good afternoon, everybody. I'm very grateful to WSSA and the USDA for giving me a chance to talk to you this afternoon about what my colleagues and myself are doing in addressing herbicide resistance with alternative chemistries in the Agricultural Research Service. So clearly there's an urgent need for new modes of action for weed control, given that herbicide resistance is evolving rapidly and spreading quite quickly. And in fact, now 23 out of the 26 known herbicide modes of action have resistant biotypes against them. And a big reason is the over-reliance in the past couple of decades that agriculture has had on chemical weed control. And the herbicide-resistant crops has only made this problem a lot worse. And the conditions that are cited most frequently as being the culprit in driving herbicide resistance is frequent applications of a single mode of action that unfortunately occurs all too often and is the leading cause of herbicide resistance. So herbicide resistance is actually a relatively new phenomenon compared to resistance to other pest control agents such as insecticides and fungicides, which resistance to them increased dramatically after 1950. And despite the fact that a lot of modes of action were beginning to be introduced in the late 30s, we really didn't see a big uptick in cases of herbicide resistance until after 1975. And it went on quite a trajectory at that point, as you can tell, so that as of today, there's 512 unique instances of herbicide resistance reported worldwide, and this is approximately equally split between dicot and monocot weeds. Some modes of action are more prone to herbicide resistance than others, and I'll go through a little bit of that later in my talk. But the first cases that, as mentioned, occurred in the mid-70s were primarily the photosystem 2 in targeting herbicides, such as the triazines, and they were widely used in corn at that time. So that propelled the uptick in resistance of that to the PS2 herbicides. And then later, during the mid-80s, ALS inhibiting herbicides resistance to that target really took off, which includes widely used herbicides such as sulfonylureas and imidazolinones. And actually, by the year 2000, this became the most resistance-prone mode of action for reasons that I'll go into a little bit later. And then finally, after more than 20 years of extensive use worldwide, they thought it was an invulnerable target, but sure enough, glyphosate-resistant biotype was identified in Australia. I actually had the chance to work on that, and then it really took off, glyphosate-resistant biotypes at that time, although most of the cases of glyphosate resistance are associated with the cultivation of glyphosate-resistant crops. So I mentioned that certain targets are prone to resistance, and ALS, acetolactate synthase, which is an enzyme involved in branched chain amino acid biosynthesis, is actually the most resistance-prone target. Just to illustrate, so to the left, this is a screen that was done at Monsanto a number of years ago, where they're using mutagenized Arabidopsis to look at the frequency of mutants. ALS, after one round saturation screen, 5 different mutants, which, if memory serves correctly, represented distinct mutations, all against ALS, were recovered. And for comparison, glyphosate didn't yield any mutants. And it turns out that the target enzyme, there's over 20 amino acid substitutions that have been identified that create herbicide-resistant ALS variant. And at the Pro 197 position, there's 12 different amino acid substitutions identified alone, just at that position. So this is the most resistance-prone target. And an important point is that these resistant variants are actually catalytically efficient. So consequently, there's very little to no fitness cost for the host that harbors these variants. So this is one of the major reasons why ALS is so resistant prone, because the target enzyme has the ability to accommodate various mutations that inhibit herbicide binding while still maintaining full catalytic activity and no fitness cost to the plant. And then there's also species that are particularly resistance prone. For example, plants belonging to the genuses Lolium, Amaranthus, Caniza, Echinoclea represent the most herbicide resistance prone reeds. And in particular, Lolium rigidum, or rigid ryegrass, is the most resistance prone species known, and has actually acquired resistance to 11 different herbicide modes of action in biotypes from 12 different countries. And perhaps not surprisingly, was also the first species to acquire resistance to glyphosate. And that was actually the case that occurred in 1996 in Australia. So why plants are prone to resistance isn't very well understood. There was an interesting study recently published by Holm and Liu that correlated wide geographic distribution, obligate outcrossing, incomplete flowers, in other words, separate male and female flowers on the same plant, wind pollination, and complex polyploid genomes, as well as large seed size, are correlated with a tendency to develop resistance. But of course, there's much we don't know at the biochemical level, at the genetic level. There's certainly other factors that predispose certain species towards acquiring resistance, and certainly there's quite a few holes in our understanding that need to be filled. So I just want to give a couple examples that underscore that resistance is a complex problem, and it's getting more complicated all the time. So to illustrate that, here's an example where two Italian ryegrass populations were identified in California, and both populations were highly resistant to glyphosate, cethoxidime, and paraquat. Both populations, high levels of resistance. And so the point being that this is referred to as cross resistance. This is unfortunately very commonplace, and which also points out the fact that addressing weed resistance problems is no longer a simple question of switching modes of action to a different mode of action for farmers, because a lot of these biotypes have cross resistance. So the complexity of the problem is ever increasing. And then another scenario that is unfortunately not uncommon. Here's a case of a tall water hemp population that was identified in Mississippi. This population does have a resistant EPSP enzyme, which does confer some level of resistance. But in addition, this population is associated with reduced uptake of glyphosate. And also, once it is taken up, its movement in the plant is severely reduced. So this is a type of so-called non-target based resistance that doesn't involve the target enzyme EPSP synthase, as well as a resistant EPSP synthase target-based resistance occurring in the same plant in the same population. These nice characterizations of these different populations was done by ARS scientist Vijay Nandula, who's actually now one of our national program leaders in the crop protection and pest management program. So another interesting case I mentioned that the Amaranthus genus is one of the more resistant prone group of plants. And one member in particular is becoming a major problem for US growers, Palmer amaranth, which is a pigweed. And it's currently one of the most problematic and economically significant weed species that affects all of our major crops, including corn, cotton, and soybean, and is a real nightmare for US growers. And biotypes have been identified in the United States with resistance to ALS inhibitors, dinitroanilines, glyphosate, HPPD inhibitors, and triazine herbicides. So this weed has just created almost insurmountable problems for growers in the States. And the most frightening and fascinating thing about this weed is it's come up with a completely new mechanism, or at least the first time we've seen this sort of thing occur in a resistant weed. And this involves extrachromosomal circular DNAs, ECC DNAs, that were discovered in Palmer amaranth. And what these are extrachromosomal DNA elements that can autonomously replicate, and they insert into various loci all across the Palmer amaranth genome, and then increase copy number in that manner. And this is not the first discovery of ECC DNAs. They're actually thought to occur in all organisms. Their normal role isn't fully understood, but they're also associated with human cancers, and they can actually promote tumor growth because they encode functional oncogenes, so they can increase the copy number of the oncogene in tumor cells. And so in the highly resistant Palmer amaranth biotypes, there could be from 40 to 100-fold increase in the copy number of EPSPS. And even though that's a sensitive enzyme, when you increase the number of copies 40 to 100-fold, even if the herbicide exposure is reducing the activity 95%, you've got 100 copies. So you're still at normal pathway flux. So this mechanism enables them to withstand normal herbicide dosages. And I also want to point out a lot of this work was done by Bill Mullen, who's a plant physiologist with the USDA, the Crop Production Systems Research Unit in Stoneville. And I believe he's the first author on a recent plant cell paper describing this work. And I encourage you to check it out. Okay, so as if there weren't enough factors that were working against us in terms of herbicide-resistant weeds, we also have to factor in climate change. And it turns out climate change can also have a catastrophic effect on the efficacy of some of our herbicides. So for example, pre-emergent herbicides are widely used in the United States in corn production, but pre-emergent herbicides require a period of rainfall after they're applied, otherwise they're not effective. And climate change will be associated with less predictable rainfall. That is one of what the models predict for how it will impact certain growing areas. So one of our research ecologists in the USDA, Marty Williams, who works at the Global Change and Photosynthesis Research Unit in Urbana, his lab performed some logistic regression models with these pre-emergent herbicides against foxtail, lamb quarter, and water hemp, and found that variable rainfall was a major driver in the loss of efficacy of these herbicides associated with climate change. So, and the reason why I bring this up in the context of this talk, not only obviously is this very concerning, but when you have suboptimal efficacy, you are exposing weeds to potentially sublethal doses of herbicide, which are the exact conditions that promote the emergence of herbicide resistance. So climate change could be another factor propelling driving the evolution of herbicide resistance in weeds and reducing the efficacy of our chemical tools. So with all these dire warnings about the current state of our mode of action, one would think that there's great impetus for and great drive in industry for the development of new herbicides. But unfortunately, the opposite is actually occurring. There have been no new major modes of action introduced in more than 30 years. As a matter of fact, most of the herbicide modes of action that we work with today were introduced between 1930 and 1985, a period where a new mode of action was produced approximately every 2 years. And as you can see, a very different case exists today. And what is widely cited as the reason for that is the exorbitant costs of bringing new herbicides to market, as well as the unpredictable regulatory environment that companies face after introducing their products. So they're very reluctant to invest in this area right now, and that could have dire consequences for world agriculture. Because if you think about it, the impact that a lack of available effective herbicides could have on the global economy, as well as world food security, which of course always affects the most vulnerable populations the most severely. This problem is every bit as significant for mankind as the loss of antibiotic efficacy due to antibiotic resistant microorganisms. So what is the ARS doing about it? And I'll go through that the rest of my talk. So the first thing I'd like to discuss is a team that is in Oxford, Mississippi, the Natural Products Utilization Research Unit. It's an interdisciplinary team comprised of Dr. Ji-Xiong Pan, a molecular biologist, Joanna Basha Herschel, a plant physiologist, Charles Cantrell, and Kumandini Meadgala, research chemists. And they are using various strategies to look for novel modes of action, including looking at determining the mode of action for novel structures that have herbicidal activity and likely to target novel cellular functions. Also looking at pharmaceuticals that may have herbicidal activity and could also make efficacious herbicides. And then, of course, the designing novel synthetics, using modeling of targets, and then a lot of effort, perhaps is more effort than in the other areas, given the location of this research unit, is looking at naturally occurring phytotoxins. That is, dipping into nature's chemical toolbox for the thousands of different active herbicidal compounds with herbicidal activity that target novel functions in the cell. And just some of the advantages of working on natural products versus synthetics include they often possess more complex modes of action. They may not have a single primary target. They could have multiple cellular targets and hence are less prone to for resistance to develop against them. Also, they've evolved in a sense that nature has refined these chemicals over thousands of years to function optimally in their natural settings. So they may require less optimization than a synthetic chemical. Of course, it's a natural product, so there'll be increased public acceptance and consequently potentially fewer regulatory hurdles to overcome when bringing them to market. And then a real key difference, when you have a natural product, that means that there's a gene and there's an enzyme that was involved in its biosynthesis. And you can also isolate those. And so you're not limited when you're working with a natural product to chemical synthesis. You, by identifying the genes, then the compound's amenable to, for example, scale-up fermentation. And you can also consider in Planta production, which I'll discuss that approach later in my talk. Also, natural products tend to have shorter environmental half-lives and so are generally more environmentally friendly. And sort of a newer realization, and I'll talk a little bit more about that, is that particularly when these natural products originate from microorganisms, a lot of times the biosynthetic enzymes are clustered all next to each other in the chromosome, and they may also be co-clustered with a resistance gene. I'll show an example of that. So, and that certainly would never be the case with when you're working with a synthetic. And I want to point out an interesting review article, opinion piece that was written by Gerwicks and Sparks a few years ago, where they're talking about the actual impact that natural products have had on the crop protection market. So their argument there, from the perspective, when you look at the top selling crop protection products on the market, the direct contribution of marketed natural products may only be 15%. But if you take into account the synthetic compounds that were either derived, modeled, or could have been modeled from natural products, this is more, the influence is more 61% on the crop protection market. So I think it's a point well taken about the significant influence that natural product chemistry has had on the crop protection market. I mentioned the advantage was you can also sometimes get the resistance gene along with the biosynthetic enzymes for natural herbicide. So in this instance, this is an interesting case on Aspergillus, and that Aspergillus produces an herbicide that targets the same pathway as the ALS targeting herbicides, that is branch side chain amino acid biosynthesis, except that it targets a different step in the pathway, the DHAD enzyme, dihydroxy acid dehydratase. Aspergillus contains a biosynthetic gene cluster comprised of AST-A through AST-D, and AST-A, B, and C are biosynthetic enzymes that produce aspartic acid, which is a potent inhibitor of DHAD. One gene is not a biosynthetic enzyme in that cluster, which is AST-D, and AST-D actually encodes aspartic acid resistant enzyme, which will counter the autotoxicity effects and the toxicity of aspartic acid. So that Aspergillus will not succumb to aspartic acid due to a susceptible DHAD enzyme, because the cluster also contains the resistance gene. So imagine this is an entire technology that's just sitting there in this cluster. You could modify them for plants, these coding regions, and also the ASD, and move the whole package into Transgenic, and now you have a aspartic acid resistant plant using a novel mode of action herbicide. So really great potential in some of these microbial biosynthetic gene clusters. So at the NPURU, the discovery flow that they typically use involves, of course, the vast repositories at the center where the unit is located, the National Center for National Products Research, which has large collections of plant, of microbial, of marine, of various animal extracts collected from all around the world. And these are used in their screening program. And also specific taxa are targeted if they have interesting characteristics that are worth investigating. Also interesting structures are examined. This is all put into the discovery flow involving model species to examine the efficacy and phytotoxicity of these various compounds. And then when they look good at that stage, they're subjected to our mode of action screening process, which first involves looking at the physiological and biochemical assays to try and nail down what pathways are being impacted by the chemical. And then also genomics are used to further refine an understanding of the pathways and genes involved. And just to show you some of the interesting leads that they've come up with. Spliceostatin is one of the interesting compounds that NPURU has identified, and it's isolated from Burkholdia soil bacterium. It's an analog of Spliceostatin A, which is an anti-cancer agent, and it inhibits splicing. And if you're not familiar with that process, just briefly, splicing is where the pre-messenger RNA, which contains a non-coding intron, is processed to the mature messenger RNA that just has a continuous coding region. And so spliceostatins inhibit this process, and molecular characterization was performed by Joanna Basha Herschel at NPURU, and she was able to demonstrate that Spliceostatin C is a strong inhibitor of both the constitutive and alternative splicing pathways in plants. Spliceostatin in the phytotoxicity test showed that Spliceostatin C is a very potent selective herbicide with a relatively low use rate, 500 mg active ingredient per hectare. And I should point out that this work was performed through a cooperative research agreement with a very innovative company in Davis, California, Marone Bioinnovations, and we've had a long-standing partnership with them and benefited greatly from this relationship. Another interesting lead is from NPURU is the compound Citral. So Citral is actually derived from key lime, and it's a constituent of the oil that's made from key lime that's actually used for organic farming for weed management. And however, the Citral, when purified from the soils, is significantly more phytotoxic than using the complete oil or any other purified constituent in the oil. And it's actually a mixture, a geometric mixture of the compound nerol and geraniol, and commercial formulations usually contain 60% geraniol and 40% nerol. Some mode of action work was done on Citral at NPURU, and just to briefly show one of the interesting results, this RNA-seq study showed 8% of downregulated genes were actually associated with nucleic acid binding. This was followed up by Ji-Chung Pan at NPURU, who subsequently determined that Citral acts by inhibiting single-stranded DNA binding proteins, such as the Worley proteins, which are a small group of plant-specific single-stranded binding proteins that are required for transcriptional defense responses. And then you can see Citral in this molecular modeling binding to Arabidopsis WHY protein. So that was a very nice mode of action study outcome. And another important lead that NPURU has come up with, so there were two furanachromones that were isolated from the Cella plant, Ames Visnagin and Khellin. And this was done by taking a dichloromethane extract from Cella and subjecting it to phytotoxicity guided fractionation. And the two furanachromones were identified, and they were not previously known to be herbicides. And this work was conducted by past and current NPURU scientists, Charles Cantrell, Kumudini Meet Gala, Frank Diane, who's no longer with our unit, and Steve Duke, who retired recently. And this was done in collaboration with some investigators from the National University of Rosario in Argentina. And both compounds were found to be phytotoxic to lettuce and duckweed. Also, they were shown to inhibit the growth and germination of ryegrass, barnyard grass, crabgrass, foxtail millet, and the Dicots morning glory and velvet leaf. Visnagin was found to be more active in greenhouse trials and exhibited significant contact post-emergence activity on velvet leaf and crabgrass. So these are obviously very important leads, and they have potential direct utility as herbicides or as lead molecules for developing more active analogs. So a patent was awarded in 2021 for this use. Now I want to switch gears a little bit, talk about a different approach, which are bioherbicides. And there's some very interesting work going on in this area in the Agricultural Research Service. So bioherbicides are essentially living entities that can act on weeds by producing inhibitors, be they phytotoxins or enzyme inhibitors, inhibiting cellular processes, or simply metabolic enzymes. But an important point is the mode of action and the identity of the specific compounds involved are, for the most part, not known. And furthermore, these bioherbicides act in concert with the target organism. So it's actually an interplay. So it involves chemistry from both the target as well as the bioherbicidal organism. So it's a complex relationship. We don't fully understand the chemistry. It certainly is an alternative chemistry, but it's essentially a black box. And so we have to focus on learning how to work with these organisms. And some very interesting work being conducted by one of our chemists, Bob Hoagland at the Crop Production Systems Research Unit in Stoneville, who's done a lot of nice work with Douglas Boyette, who's a plant pathologist, also in Stoneville at the Biological Control of Pests Research Unit. And just to quickly summarize this really interesting work. So they have one bioherbicidal fungus, Myrothecium verucaria, that they tested against the glyphosate-resistant Palmer amaranth, which, as you know, as mentioned, is one of the worst weed problems facing American growers. And you can see here, when treated with glyphosate, you can see the glyphosate-resistant Palmer amaranth is the bottom panel and wild-type plants are the top panel. And you can see very little effect of glyphosate on the resistant Palmer amaranth, a fairly significant effect of the bioherbicide. But when you combine the two, you get this synergistic effect and you get almost complete inhibition of the growth of the herbicide-resistant biotype. And in fact, Bob and Doug identified this as a synergistic interaction. And so when you have a synergistic interaction, you get a much more powerful effect than either component by itself. And so this is one novel and very promising alternative way of controlling some of these very difficult herbicide-resistant biotypes. And another point is when you identify a synergistic interaction, that means that that typically requires relatively low levels of both components. So there's also an economic advantage to working with these kind of synergies. And so they're doing a lot of interesting things in this area that's impactful to the herbicide-resistant weed problem, and you should check out some of their work. Okay, so another area that the ARS is involved in herbicide-resistant weeds is plant-incorporated pesticides. So plant-incorporated pesticides, the most well-known example are the Bt toxin-producing crops, which have had major impact on insecticide use, reduce the amount of synthetic insecticides required by growers, and also has had significant cost savings for growers who don't have to spend so much money on chemicals. So we feel that plant-incorporated herbicides could have the same impact on agriculture. However, at present, there are no plant-incorporated herbicides on the market, and so we are working to address that shortcoming. The fact is that nature has been in the business of plant-incorporated herbicides for many eons, because this is one ecophysiological aspect of plants is that many plants do produce herbicides to take out their competitors. And these chemicals are called allelochemicals, and this is a major field of study, allelopathy. And it's thought of as sort of a chemical warfare that occurs between plants that are competing for limited resources such as light and water and nutrients, and are able to take out their competitors by producing phytotoxins that they release into the environment. And so some of the more extensively studied examples include the mummy-lactones that are produced by rice plants, the benzoxazinoid compounds that are produced in a number of grasses. There's also gramine and hordenine produced by barleys, scopolectin from oat. And one of the most extensively studied allelochemicals is the compound produced by sorghum bicolor that I'll talk a little bit more about, Sorgoleone. So Sorgoleone is a benzoquinone. It's an amphipathic molecule, as you can see, with this polar head group and long hydrocarbon side chain. And interestingly, it's only found in the genus Sorghum. And we actually did a survey a number of years ago where we took samples throughout the grass family, and only members of the genus Sorghum, Sorghum bicolor, shattercane, Sudan grass, Johnson grass, you could find some level of Sorgoleone, but once you go outside of the genus Sorghum, you don't find a trace. And Sorghum bicolor is a prolific producer of this compound. The entire root system is coated with Sorgoleone. And in fact, if you look at the exudate, the root exudate from a sorghum plant, in some cases it can be up to 90% weight to weight of the content of the exudate. And you can actually dry it down and use it directly as an analytical standard in some cases, which is very convenient. And the reason why, one of the main reasons why it's targeted as for plant-incorporated pesticide development is because it's such an effective herbicide. It's a potent herbicide, and it has efficacy against numerous economically important weed species. And it's also thought from the research done in the allelopathy community that it could be the major factor that confers allelopathic activity to the sorghum plant. In other words, its innate ability to fend off weeds could be largely attributable to its production of this chemical. And which it's also an interesting system, a difficult system to work on because it's only produced in root hair cells. So you have to learn how to work with root hair cells. And so we've been doing that. And we had none of the genes, of course. So we've been diligently working in recent years to identify all the genes required for its biosynthesis from the ubiquitous precursor palmitoylea-CoA, which is essentially found in all plant cells. And if you add 5 genes, you can produce Sorgoleone. And so we've been working to identify all these genes, and we managed to isolate all of them and biochemically characterize them and fully elucidate the pathway. And so now I just want to mention that it's been added to the Metaseq International Biochemical Database as a new pathway. And I'm very happy that our unit and the USDA was able to contribute to fundamental knowledge of plant secondary metabolism in this manner. So luckily, there was a group a few years before we started that had figured out a very clever, simple way to isolate root hair cells. They were making protoplasts, and it was simply a matter of putting the root systems in liquid nitrogen, then filtering them and allowing the nitrogen to evaporate. And then you, once the nitrogen evaporates, ideally you have a very purified root hair preparation, which can then be directly used for RNA isolation and making library sequence analysis. And then once we had the root hair transcriptome generated, then we added a secondary screen so that we could look for genes only expressed in root hair cells to generate the candidates for our enzyme work. And this approach turned out to be successful, and I'll just quickly go through some of the highlights. So Ji-Chung Pan in our group worked on the desaturase, and he was able to characterize DES2 and DES3, which convert the ubiquitous precursor I mentioned, palmitoylea-CoA, and they act on it sequentially to produce a delta 9, 12, 15, 16, 3-CoA, which is actually the entry molecule for Sorgoleone biosynthesis. So that was very nice work. Then my lab was involved in characterizing the methyltransferase. OMT3 only accepts or vastly prefers alkyl resorcinolic substrates and can also act on the physiological substrate pentadecatrienol resorcinol. And so that methyltransferase participates in the Sorgoleone biosynthetic pathway. And then we also were involved in, my lab was involved in working on a novel type of type 3 PKS enzyme that had actually never been identified in any organism before, which unlike enzymes like chalcone synthase, this PKS uses linear starters or acyl CoAs, and then can, through iterative condensation reactions, build the aromatic head group or alkyl-resorcinol product. And so these are actually referred to as alkyl-resorcinol synthases. And so we identified 2 genes in sorghum that perform this reaction. And they only accept linear so-called starter molecules and don't work at all with substrates typically used by other type 3 PKSs, such as benzoyl-CoA, et cetera. Okay, and then the last piece of the puzzle, this work was also done in Ji-Chung Pan's lab, the identification of CYP71AM1, which performs the last steps in the pathway, the dihydroxylation of the pentadecatrienol 3-methyl ether to produce dihydrosorgoleone, which is actually This is actually the hydroquinone form of Sorgoleone. It undergoes spontaneous autooxidation once secreted into the soil to form the more biologically active, redox active benzoquinone. So once this work was completed, we then had all of the genes required to go from palmitoyl-CoA down to Sorgoleone. So the tools were in hand to engineer plants to produce this compound. And I just also want to mention that the alkyl-resorcinol synthases have also given us a means to actually manipulate Sorgoleone levels in sorghum. So we've actually developed germplasm that completely lacks Sorgoleone through RNA interference by targeting the alkyl-resorcinol synthases. And we can also do, here's an early simple result using the enhanced 35S promoter, just overexpressing one of the synthases. And in event 84-1, we actually got a 60% increase in Sorgoleone. So this appears to be a potential control point in the pathway, and we can use it to manipulate levels of Sorgoleone, which in and of themselves are potential products. And I also want to mention that there's a lot of interesting things that can be done with these mutants. And if anybody's interested in using them to address questions that aren't directly overlapping with our work, of course, I would be happy to share them or collaborate with you, so please feel free to contact me. And likewise, we've had to generate some promoters which weren't available in the literature. Since Sorgoleone is expressed in root hair cells, we needed strong promoters to engineer, do metabolic engineering work in other crops with these genes. So we isolated some excellent, very specific, very powerful promoters that are active in both monocots and dicots. And beyond our project, these promoters also have utility for a lot of other areas, such as enhanced nutrient acquisition, plant pathogen interactions, bioremediation. So if anyone has a use for these types of elements, please feel free to contact me and I can provide them. Okay, so obviously we want to produce crops that make Sorgoleone. And so we're in the proof of concept phase with just using the biosynthetic enzymes. And so we're working with the Wisconsin Crop Innovation Center to put in this 5-gene vector into corn, soybean, and wheat. And we've also have some transgenic rice plants. We don't know how different species will respond to producing this toxin. So we're using a multi-species strategy. And if we have success in this area, I feel it'll be a very important advancement in the plant incorporated protectant field. So, but we're not quite there yet, because it's not enough to just produce the compound. You actually have to transport the compound to its site of action. And in the case of Sorgoleone, that's out in the rhizosphere. So we still need the transport components that are required. But fortunately, Sorgoleone is a type of lipid. It's referred to as a phenolic lipid, and there's a lot known in the extracellular transport of lipid precursors in plants from some fairly well-studied examples. So, for example, the cuticle on the leaf surface, it's known, it has a major lipid component to it, and these lipid precursors are actually transported outside via an ABC transporter that's then handed off to lipid transfer proteins, which then help move it to its final destination on the leaf surface. And likewise, during the development, superization of the root vascular steel, the endodermal cells produce the lipid-rich precursors for suberin, which are then moved out of the cells through ABC transporter activity, handed off to lipid transfer proteins, where they're then deposited in the Casparian strip. And another example is in the developing pollen, the exine coat, which is largely sporopollenin, which is a lipid-rich compound. The precursors for the pollen cell wall development are produced in the tapetum or the tapetal cells, which produce these lipoidal constituents that are moved out into the locule via ABC transporters and then assisted in moving to the final destination with the assistance of lipid transfer proteins. So there's well-studied examples that we can model our own work on and propose that a similar process occurs in sorghum root hair cells, where the Sorgoleone is moved out of the cytoplasm with the assistance of ABC transporters, and then moved to the rhizosphere with the assistance of lipid transfer proteins. And so we're actively working on identifying the components in sorghum that carry out that activity. And then I want to bring up one more, I think, really important point about these transporters is that they are what are referred to as efflux pumps. And efflux pumps play a major role in nature in autotoxicity avoidance. So a lot of this, a lot of the research has gone on in microorganisms, but higher organisms, eukaryotes, conduct the same process using these efflux pumps. There's three major types of efflux pumps that you'll find in plants and other eukaryotes that belong to the major facilitator superfamily, multi-drug and toxic compound extrusion or mate superfamily, and then the ABC type transporters. And all of these are involved in various systems in extruding toxic compounds from cells that create sublethal conditions within the cytoplasm so that the organism does not succumb to the toxic effects of the compound that they're producing. And so that, in a sense, confers resistance to that organism, to what the compound they're producing. So we can then envision the case of Sorgoleone. And I want to point out, I think I mentioned that sorghum plant is a prolific producer of this chemical. I mean, it is pumping it out. It's constitutive, so it's what's referred to as a phytoanticipant. It's not a phytoalexin that's released upon some stimulus. So it's not, it's produced in massive quantities continuously. So storing it would not be practical. Metabolically inactivating it, well, you'd still be faced with the problem of where do you keep it? So obviously what the plant needs to do is get rid of it. And that's undoubtedly what it is doing. And so these efflux pumps and likely an ABC type transporter will play a critical role in autotoxicity avoidance. Moreover, this will complete the technology so that these pumps will also protect other crop hosts that we move the Sorgoleone biosynthetic pathway into. We will also provide them with this pump, which will confer resistance to Sorgoleone. So that is the final piece of the technology puzzle. And we're well along in this area. We have transfer protein and transporter candidates, used a genomics-based approach. We found, interestingly, there are clusters of lipid transfer proteins that are root hair specific on 2 different chromosomes in sorghum. And to add a little firepower to this project, we've struck up collaborations with Texas A&M, Sakiko Okamoto, who's actually an expert in plant transport, and also have a new cooperative agreement that we're initiating with the Donald Danforth Plant Science Center, who have significant expertise in cask CRISPR studies with sorghum, which is not trivial, and we're very pleased to have that kind of guidance. So I just want to take a second to mention, obviously all of this work on the Sorgoleone project required a lot of people, and I've just been very fortunate to have so many talented colleagues and collaborators on past projects and ongoing projects that made the progress that we've had to date possible. And with that, that's all I had to say, and I'd be happy to take any questions at this time. Thanks a lot for your attention.
Yeah, thanks, Scott. That was great. We do have time for a few questions, so I'm going to just ask you, Scott, if you can address. I've got a question on the idea of resistance to, I guess, the natural-based products as they are naturally occurring, and this idea of coevolution with, the crop or the, where they're being used. Is there any potential for this idea of resistance to these novel mode of actions, where they're present in wild populations? Is that a concern at all?
It is known. It's not. I mean, it's certainly, yes. These compounds arose in natural ecosystems, and so it has a specific ecophysiological role, and there will be, and actually chemicals like illegal chemicals play a major role in shaping ecological communities because of their toxicity. So yes, you get co-evolution, you get relationships between species. Overall, does that invalidate them as good candidates for natural product herbicides? Absolutely not. I mean, time and again, you find examples. And plus, we're not targeting, I mean, the weed species that are on, commercial growing operations are very different than the, the organisms that reside within their natural community structures. But certainly, yes, that, coevolution, various levels of tolerance and susceptibility in natural communities does exist. How much will it impact their efficacy in agricultural settings? I mean, to some extent, perhaps, but just from what I've seen and from the examples that we've worked with, they seem to work very well against the economically important weeds in many cases.
Yeah, a little bit different question here from, this is in the, just reading the chat about, have you had any experience with eugenol-based herbicides? Any experience with those?
Now, again, I've been working on the Sorgoleone project, so to ask me specifically, the answer is no. Has anyone in our unit? Geez, I would have to, follow up on that.
Yeah.
I'm sorry, off the top of my
Yeah,
head.
no, that's fine. And I would, again, anyone in the audience, if you have specific questions like that, feel free to ask Scott. We have a question on the Sorgoleone fit into systems where, again, we're getting kind of specific here. So, just you take that into consideration, where there's Striga asiatica. It's been shown to be a problem since it's shown to stimulate its germination. So again, any thoughts on that? And maybe the bigger question is, do you see this fitting into systems, outside of sorghum system? I mean, does this apply to the broader cropping systems in general?
Well, I guess what I was, if I'm understanding the question correctly, what I was trying to point out toward the end of my talk is we're trying to make this technology so that it has built-in resistance that it will confer to whatever host we put it into. And I thoroughly believe, and my transport colleagues are in agreement with me, that these transporters will be key to making this technology viable. And whether or not that is the entirety of the mechanism that sorghum plant itself uses to avoid autotoxicity, by increasing the expression level of this transporter, we can make it super, we can use the right promoters and genetic elements to raise the level of expression of it much above what sorghum plant normally expresses it at, so that it will most certainly pump out anything trying to diffuse back into the host that's producing it. And so it really feels strongly that we will have a pretty bulletproof autotoxicity avoidance mechanism built into this technology.
Yeah, I just want to end with kind of, so you said, you've been at this for 32 years, and I'd like to know, as you've been kind of exposed, are you seeing more of an acceleration of this kind of approach and technology, more than you have, I guess, when you started out? And maybe there's an obvious yes to that, but I'd just like to hear your thoughts about, when you first started and right now. Are we seeing things really accelerate with how this research, this direction is going?
I really feel, well, of course, when I started out, and given, as mentioned, that I've been doing this for 30 years, obviously, when you go back 30 years and think about the techniques and the technology that was available to us at the time versus, the explosive advancements in genomics and sequencing technology, we now have the ability to do things that, we couldn't even conceptualize 30 years ago. And add on another 20 years to that, because molecular biology and genomics, just the growth in this field is so dramatic that I'm very excited about, especially some things that were brought up in some of the other talks, nucleic acid technology. I mean, I think technologies, and it'll become easier and easier with genomics to come up with new pathways to do plant incorporated pesticides. Also the gene editing technologies, there have been dramatic breakthroughs. So our ability to add these genes without actually doing genetic modification is improving. So I actually, we do need new modes of action, absolutely. I mean, that's critical. But I also think that genomics and nucleic acid technology, I don't know, 30 years, I don't know exactly what the timeline is, but weed control, I think you'd be shocked at how we're going to be doing weed control 40 years from now, with designer nucleic acids. And I mean, I really think the technology that are going to be developed in the next few decades is going to be pretty astounding.
Yeah, and I think you touched on that with Dr. Mullen's work, looking at how do we take some of these traits and actually put them in crops. So we're developing crops that are more resilient, more resistant to some of the things that weeds already, are kind of can do already. So I think that's where this molecular genetic kind of approach and techniques are really going to help us advance and see a lot of that happening. So anyway, I just want to say thank you. I know that there's some other questions here, but I'd encourage folks to contact either myself or Dr. Baerson to follow up. Really some exciting research here. And I would also say that we're actually looking to hire a weed ecologist looking at some of those genetic issues in the Southeast. So just to push that a little bit for you, those of you who are looking for a position, ARS is actually hiring. So, feel free to check that out. But anyway, thank you, Scott, for your time today. Appreciate you sharing your research, really exciting stuff. And I just want to say next week, we're going to hear, again, something different. We're going to hear about the spread and distribution of invasive plants. So we're going to move to our final theme of impacts. So hopefully you all can join us. Like I've been saying, every week, same time, same place, and we'll see you then. So thanks, everybody. We'll talk to you real soon.
Thank you.